Okay, good morning, good afternoon, good evening, good late evening, depending on where you are around the world. I'm Prieto-Alhambra. I'm a professor of Pharmacoepidemiology at Oxford, and one of the colleagues who joined the OHDSI community not that long ago, although I feel like a veteran by now. I am delighted and honored to moderate this panel discussion. I hope you enjoyed the presentations. We have a great roster of panelists who I'm going to introduce very briefly. Catherine Cohet is an expert in pharmacoepidemiology and real-world evidence methods, and she's one of the colleagues who is working tirelessly on COVID vaccine safety analysis and surveillance within the European Medicines Agency in the Data Analytics and Methods Task Force. Welcome, Catherine, and thank you so much for your time. Richard Forshee is the acting deputy office director at the Office of Biostatistics and Epidemiology at the US FDA CBER, Center for Biologics Evaluation and Research. He's got over 20 years of experience in health policy research. And again, I am delighted to welcome you to this panel, Richard. Thank you for your time and thank you for joining us. Third, we have Joanne Waldstreicher, the Chief Medical Officer of Johnson & Johnson. She has oversight across pharmaceuticals, devices, and consumer products for safety, epidemiology, clinical and regulatory operations, and technology and R&D policies. Thank you for joining us, Joanne. And last but not least, we have Professor Magda Sobieszczyk, who is a professor of medicine and chief of infectious diseases at the Columbia University Irving Medical Center in New York. Magda has an amazing portfolio and CV of publications and work on HIV and SARS-CoV-2 infections, and specifically on vaccines, monoclonal antibodies, and pre-exposure prophylaxis. Thank you for joining us, Magda. Okay, so what I thought we could do is I have a couple of questions that I can use to break the ice, and I can start with those. And George Ripsack, who I didn't introduce, but I think most of you know already, he's the head of the Department of Biomedical Informatics at Columbia University. He will help me with moderating questions that might come from the chats, if that's okay, George. Okay, so my first question, and when you answer, please, very welcome to have any immediate reactions or any thoughts that you might have on top of the answer to my question. The first one is about all the methodological research that the OHDSI community has contributed to inform the COVID pandemic. From your perspective, what of these methods research has been useful, and how could it be even more useful if possible? I might start maybe with Catherine.
Yes, thank you, Danny, and thanks for the introduction. So to answer the first question, well, I think it's obvious for everybody on this in this meeting that methods research is crucial to understand the limitations of observational data that are used in regulatory decision making. So I was really impressed by all the presentation. There was a lot of insight into the methods, and a number of examples have been given by the speakers of how OHDSI work has been used for discussion at the agency. And just to name a few, the work on hydroxychloroquine and the risk of mortality that led to a warning for healthcare professionals treating COVID patients, or very importantly as well, the work on background incidence rates, not only for predefined adverse events of special interest, but also to support emerging signals, such as the signal for thromboembolic events. I'm referring here to the talk by Xin-Tong Li. I mean, I was particularly interested in all the discussions around how to best generate and interpret background rates, and this is a backbone for any discussion around signal management, which usually start with observed to expected analysis, and that in the continuum of the signal evaluation process. So it's really important to understand this data. And there was also an example, I think it was by Rupa, about Guillain-Barré disease, which is a bit a usual suspect in vaccine safety, if you will, but we need to be prepared for methods to address emerging unprecedented signals, and such as, again, the thromboembolic events or multi-inflammatory system syndrome. So just to finish, I think this methodological work will probably help even more moving forward as the vaccination campaigns continue to be further deployed in new population groups, and also how we will leverage lessons learned. And I think we had a good illustration today already of the experience that is accumulating. And I'll stop here. Thank you.
Thank you so much, Catherine. I think it's it's quite fundamental how we phenotype these events and these outcomes and how we use them in real world evidence. Richard, can I invite you to come in and tell us your thoughts on methods research that the community has contributed and how that has been useful or could have been even more useful?
Yes, certainly, Danny. And I largely agree with what Catherine said earlier. I first just want to say these presentations were really fantastic. A few things I want to emphasize is that the work here is really providing some fundamental empirical data on a lot of the difficult choices that we need to make when we're designing any epi study. And the work on anchoring dates, I think, was just a really good example here. That's something that we always consider in protocol development, but I had never seen such empirical data to help inform those choices as what was presented earlier today. I also want to compliment the team on the way that they're approaching this research. One of the presenters mentioned that this is following the principles of open science, and it certainly is. There's so much of the work is so much work is done to make sure that everything is transparent and reproducible, from the publication of the protocols in advance to providing code for people to reproduce and follow up on those results. So it's a really good model of how to do this kind of work well. And I think the last thing I'll mention is just how thorough each one of these projects are. The number of analyses and the combinations that are explored in each one of these efforts really go beyond what you typically see in epidemiological studies. And again, I think we're learning a lot from it, and I think it provides a very good example of how to do high quality epidemiological research using observation data. And I think I'll stop there for now. Thank you.
Thank you, Richard. And yeah, I think that's, again, one of the main strengths of this community is the effort towards open science and reproducibility and transparency. Joanne, can I please ask you to let us know your thoughts on this topic as well?
Sure. Just a little bit of background. The Office of the Chief Medical Officer, which I lead, is within J&J, but it's a global and functionally independent group responsible for leading both medical safety as well as epidemiology, as well as some other responsibilities. But our functional independence, the fact that we don't report into commercial or R&D or other parts of J&J, really empowers the safety teams and their leaders to make decisions free of any potential commercial or development bias. And since we've been established in 2013 within J&J, we've worked to advance evidence and science-based decision-making that's patient-centered. And real-world data has been front and center to all of our efforts. And that's why we as J&J and ACMO, as we call ourselves, is one of the founding partners of OHDSI with Columbia. And I have to say that since the pandemic started, the work of OHDSI has never been more important, I think, globally, just in general, but also to our work in J&J, with our highest priority during the pandemic to work towards combating COVID-19, but really to ensuring effective and uninterrupted monitoring and evaluation of the safety of our products across the enterprise and across the globe. And the methodologic work has played a really crucial role in helping us establish our strategy for using real-world evidence as part of our own safety surveillance activities for our COVID vaccine. It's critical for us to know that the evidence that we're using is reliable. And this speaks to the importance of the foundational science that can build trust in real-world evidence. And understanding the background rates of adverse events is a significant part of our work towards vaccine safety surveillance. And to that end, OHDSI's work to generate a resource of background rates was extremely helpful and used many times by our teams. So was the earlier research on the methods for estimating the incidence and also the sensitivity of those results to the different design choices and phenotype algorithms. And when using the data for safety surveillance, we had to understand how well those methods work for safety surveillance, what trade-offs might exist between the alternative approaches. So the work that Martijn Schume presented and Faiza Arshad provided interesting insights on the potential methods to identify those safety issues as soon as possible, and also insight on how to do so in a manner that reduces the number of potential false positive findings. The patient-level prediction that Jenna Reps presented is another exciting area, taking us beyond characterization of just observing the events or estimating the effects of exposure, and helps us better understand which patients have the different risks. And her work examining how machine learning algorithms can be applied across different populations just provides us some promise that these approaches can someday inform our portfolio of safety analytics activities. So again, hugely important to our day-to-day work, giving us a lot of confidence, a lot of understanding of the sensitivity of the different methods, and a lot of resources in our toolbox for doing safety surveillance.
Thank you so much, Joanne. Magda, are you okay to come in and let us know your thoughts on methodological research? I'm going to maybe ask you to specifically, because I appreciate that you are a person who also has led a lot of clinical trials, not only observational research. And because you are here, I assume that you are one of the people who are trying to make those two worlds converge, right, and diverge as we see sometimes. If you can tell us your reflections on methods work and how we can try and bridge the trials and real-world evidence worlds, I think that would be great.
Yeah, no, absolutely. Thank you. And, I have the advantage, I think, I agree with everything that all the other speakers have said in terms of the kind of the incredible importance of this type of methodological research to sort of kind of harness the observational data to as you're saying, inform both, I think, thinking about kind of both clinical practice, but also kind of inform design and implementation of clinical trials. And I think one, I was really struck about kind of, about sort of the very thoughtful presentations, kind of about the careful selection of cohorts and definition of cohorts, and also recognizing limitations, right? That there's still some work to be done, which we can talk about later, because I think that is critically important when we are thinking about designing an interventional study, like what cohorts do we go into, what populations do we focus on? So I think sort of being kind of very thoughtful about the methodological research and how cohorts are being selected to define incident rates, define sort of, background rates, I think it's really important. And then also kind of the definition of the disease phenotype. I was really impressed kind of with the thoughtfulness with how this is being approached. And also kind of thinking just about the challenges of picking up rare signals, right? That rare signals, one can say, oh, these adverse events are incredibly rare. Why do we care about them? But when we are sort of deploying vaccines on a incredibly large scale, and also living at a time when everything is being amplified in the public discourse, and clearly, patient safety is a huge issue. Kind of these rare signals are incredibly important. So kind of thinking about how this type of research and looking at observational data, how it can help us kind of thoughtfully pick up these rare events and recognize that there may be limitations, how we pick them up, and how is incredibly important. So these are just some general thoughts, and I'm really excited to kind of dive into the future further discussion.
Thank you, Magda. Okay, my second question is more about the clinical evidence or the clinical knowledge that has been generated. I guess you enjoyed, as I did, listening to speakers from all over the world discussing the work we did during the COVID lockdown, when everybody was emotionally affected and professionally affected, and we had to figure out how to unravel a new condition and create knowledge that could potentially be useful, and also at a time when there were other other initiatives launched by many stakeholders trying to do similar things. So I'm wondering, Catherine, if you want to tell us a little bit about your thoughts on clinical evidence or clinical knowledge that has been generated by the community, and how we could have been doing better if possible.
Thanks, Danny. Well, I think I've already touched a little bit on methods-based preparedness, but I think here clinical evidence also helps preparedness and response. And Peter Reinbeck, in the beginning of this plenary earlier today, mentioned how EMA has supported a number of studies using the OMOP common data model, which in turn informed decision making and particularly supported discussions at PRAC, our pharmacovigilance committee. Another point, I was just listening to Magdalena, and I like this discussion about trying to merge the two worlds, and I think the type of clinical evidence that has been generated has also allowed to inform maybe not the design, but certainly the interpretation of randomized control trials. And this is important not only in the post-authorization phase, as more and more trials are being evaluated for further indications of the vaccines, but it's also important more widely and beyond COVID-19 vaccines. I think it's also important for pre-licensure and peri-licensure decision making. In terms of more specifically the clinical evidence that has been generated for COVID-19 vaccines, I think probably most people in this meeting are aware of the signal of TTS, thrombosis with thrombocytopenia syndrome, which was a completely new syndrome. And it's been extremely challenging to come up with case finding algorithms, the right phenotypes. And this evidence, particularly coming from the background rates, has been really important at EMA to enable the contextualization of the benefit risk of some of those COVID-19 vaccines, and in turn this helps supporting decision making in Europe at national level in terms of recommendations for a second dose, or also in terms of which age group might be impacted. And lastly, in terms of the clinical evidence, I was particularly interested in the last talk this afternoon about effectiveness. I don't know if everybody's aware, but effectiveness is actually now part, or generating effectiveness data is now part of the extended mandate of the EMA, and recently a lot of methodological challenges have emerged in the context of discussions around duration of protection, waning immunity, need for booster doses. So I'm really curious to see how such evidence can be generated using the OHDSI community network and more generally secondary data. And I'll stop here. Thanks.
Thank you, Catherine. I'll take some notes on your wish list for the community. Richard, do you want to comment?
Sure, I'll add a little bit to all of the great comments from Catherine. I think I'll start by saying I've always viewed real-world evidence and clinical trials as being complementary sources of information. The RCTs will remain the gold standard for determination of effectiveness of the efficacy of a product. But there's always going to be a lot of questions that are still left over after a trial. There are subpopulations that would have been excluded from the trial. We don't have the long-term follow-up. So there's always going to be many questions where real-world evidence can round out what's been learned from the clinical trials, and sometimes answer questions that would just be impractical for a clinical trial to do. So I think it's really important to view these as complementary sources of evidence for the decision making, for the decisions that we have to make. In terms of where I've seen real world evidence already contributing on the clinical side with the COVID-19 pandemic, I think the earliest places that we saw the contributions from real world evidence were in natural history studies. And I know our team did one of those using our Medicare data for the over 65 population in the US. And OHDSI conducted a number of natural history studies. There was so much that we didn't know, particularly about the health conditions and other factors that might put people at higher risk. These early studies were critically important to provide some more information there. As time moved on, we got to a point where the real world evidence was providing very important information on safety issues, such as the TTS issue that Catherine mentioned. And also in the United States, we were running rapid cycle analyses on multiple claims databases to track about 15 adverse events of special interest for any early indications of an increased risk. And now I think we're turning to the point of looking at the information we can get on effectiveness of the vaccines, and in particular looking at questions such as waning effectiveness, effectiveness against variants. And some of these are going to be very difficult questions to disentangle, but they're important questions, and I think observational data is where we're going to get some of the earliest and best information on these. I'll go ahead and stop there. Thanks.
Thank you, Richard. I will add those last two to the wish list as well, and I'll try to recap later and see where we stand. Joanne, do you want to comment on clinical evidence generated by the OHDSI community on COVID, and if you have anything to add to the wish list?
Sure. Well, first, I just want to say I completely agree with the previous two panelists. The OHDSI work really provided valuable insights throughout the pandemic and throughout our own efforts across COVID for both therapeutics as well as vaccines, and that directly impacted our work. Charybdis provided really important insights into the disease, natural history, and treatment utilization that guided our thinking, as well as providing context to some of the outcomes for people with COVID and putting into context some of the outcomes with the vaccine so that we can understand the relative risks for people with COVID for having certain outcomes, as well as the relative risks for the vaccine, and put that all into kind of a benefit risk perspective. I have to say the COVID vaccine adverse events of special interest background rate, and I mentioned this before, but that was really important and something that we have and still use actively in our ongoing safety surveillance efforts. Seeing rates across different databases, stratified by age and sex, et cetera, really provided context to help compare what was observed in our vaccinated patients with what may be expected. And in fact, for those of you may have seen, I actually used results from the OHDSI study in our presentation to the CDC's Advisory Committee on Immunization Practices, or ACIP. It was so important to our efforts there. As we continue to learn more about COVID-19 treatments and vaccines, real-world data will not just continue, but I think will increase in its importance to understanding the effectiveness of the vaccines, and as the other panelists have mentioned, really important because it's always complementary. Real world data is complementary to clinical trial data. We've had extensive clinical trial data, placebo-controlled clinical trial data. That window is closing, where it's very, very difficult for certain vaccines and therapeutics to do any more placebo-controlled trials. We're going to have to rely more and more on real-world data to understand the effectiveness, whether there's waning immunity, what the impacts of the new variants on effectiveness, and new safety issues that might arise, we don't know, as we start looking at different regimens, boosters, et cetera. So I think real world data will become increasingly important, and the work of OHDSI will also become increasingly important.
Thank you, Joanne. I entirely agree with what you said there. I think there's going to be a continuum between the data that we've learned from the RCTs and what we will be learning from real world evidence. That's going to be very important, that continuum is smooth rather than shaky. Magda, maybe to keep you a bit on your toes, I'm going to ask you to take off your academic hat and put on your excellent clinical academic, but clinician hat, as the chief of infectious diseases in a hospital, in a place of the world where there was a lot of cases of COVID-19 during the first wave of the pandemic, right? What evidence did you need at that stage? What evidence did you get, and what evidence out of the evidence produced by OHDSI, but also potentially by other sources, was important?
Yeah, no, thank you for that question. I was also sort of kind of reflecting on kind of taking a step back or 5 steps back to exactly to kind of to put on that kind of that clinical hat, both in terms of, giving guidance to clinicians and also talking in conversations with patients, right? What are kind of some of the key pieces of information that I think OHDSI has and others have just been able to provide. I think kind of reflecting back on that time, this was certainly a time of incredible uncertainty, and we kept using the term that we're operating in a data-free zone, relying only on in vitro data and information kind of extrapolations. And I really enjoyed the presentation on how the rise and demise, so to speak, of certain antimicrobial or antiviral agents sort of over the last 12 months. It was incredibly powerful to sort of do. That presentation was beautiful. So yes, I think just sort of these types of data that were generated by this group, by OHDSI, was incredibly helpful in providing a clinician as well as a sort of a patient advocate with real information about kind of identifying patients at greatest risk of adverse outcomes. You may have a early in vitro data or early results from clinical randomized trials, but it really is sort of being able to kind of fall back on that observational and real world data about the risk of HIV infection or higher BMI kind of on certain clinical outcomes. It was incredibly important and incredibly powerful. And I think I was reflecting on how very frequently we were changing our local hospital guidance almost on a weekly basis, really in reaction to the data that were being generated by investigators who were presenting here today. And it's, I think, sort of what was required at that time was an incredible level of nimbleness to be able to kind of incorporate the data that was emerging and really translate it into clinical practice. And I think what was very powerful is sort of knowing that OHDSI was able to kind of knit large data sets from many, many different parts of the world and really kind of do it in a systematic and methodologically sound way. And that was sort of critically important because, deployment of randomized clinical trials, as I think Richard, you and Joanne have said, is not is not always possible. It may not be possible in the future. So I would also want to look into kind of in the future, sort of thinking about how this group can help us sort of evaluate the validity of certain surrogate markers that will have to be used to evaluate effectiveness of either certain therapeutics or even vaccines, because we will not be able to have sort of these, beautifully designed placebo-controlled trials in the future, but we may settle on selection of certain surrogate markers as proxies for effectiveness, for efficacy, for outcomes. So how do we then use these surrogate markers that are being then utilized in the real world to evaluate long-term effectiveness of certain therapeutics? And I think that's kind of another maybe call to this group to sort of think about how to do that in an innovative way.
Thank you, Magda. Just to be fair and to keep everyone on their toes, I'm also going to introduce a new question in the panel that came to my mind as we were speaking about clinical evidence. As an academic, contributing to these efforts, and as a clinician, we struggled a little bit as a community. I hope some will agree with me, to get our data published in top journals. We saw some journals prioritizing speed over quality sometimes, and I wonder how do you get evidence? Where do you get evidence from for regulatory purposes, for your work at J&J, Joanne, and for clinical management of patients? How did you do that during the pandemic, and how do you think that's going to shape how we do things in the future? Are things going to change, or are we going to keep screening medRxiv and SSRN for results? Does anybody want to kick off and answer that?
Okay, maybe I can just make a few comments on the publications and where the data come from. And we evaluate data as it's generated, and a lot of data is coming from medRxiv and preprints and all that. And of course, this is taken up by the public, by the media, and very often we have to evaluate this data because it's out there, and we have to have an opinion, and it's discussed a couple of times a week in the EMA Pandemic Task Force, and it's a bit challenging because you might have several iterations of a paper, and then eventually the peer-reviewed approved paper is out and data might change. So it's a challenge, yes.
I can only agree that, Richard, why don't you go next? Sorry.
No, please, Joanne, you go first.
No, I was just going to say it is a real challenge, and we do the same thing where we have our team here, many of whom evaluate the papers as they come out on medRxiv. Many times, especially if it affects our own vaccine, we'll try to quickly do an analysis ourselves if we have the tools. We leverage the OHDSI tools a lot, see if we can confirm the finding. But it's a very difficult situation when the OHDSI papers, as you're saying, Danny, are not accepted right away, or there are other publication. There's differential publication, I would say, where speed is prioritized. And I think it sets up a very difficult situation. But I think also that the world of science has learned a big lesson in the past year and a half. I won't mention any specific situations, but I think we all know, there were some situations with some real world data that came out, and there were some good questions raised. I think the same thing happened with clinical trials. So I think that one thing that COVID has shown us is that we have to raise the bar scientifically. That's a good thing. And sometimes speed is not always the priority. And I think as OHDSI keeps showing that more and more, and other good researchers keep showing that more and more, the playing field, as we say, will be more level over time, and good science will prevail.
So I can provide a few comments here.
Please.
I do need to begin with a disclaimer since we're getting near regulatory policy. This is an informal presentation and my comments represent my best judgment, but do not bind or obligate the FDA. That out of the way. I will say that it is a real challenge to try to use real world evidence for regulatory decisions, because we have very high standards for the quality of evidence that we are going to consider. Some of the things that go a long way towards making real world evidence more reliable for regulatory decisions. First of all, we need the protocols to be publicly posted prior to the start of the analysis, so we know we aren't seeing the result that turned out the way the sponsor wanted. It's also really important if a potential sponsor is conducting a real-world evidence study that they want to have considered as part of the regulatory evidence, talk to your regulatory agencies early on in the process, so you can get to some consensus on what the expectations are going to be for everyone. The results also need to be reproducible, so make sure that the work that you've done is something that others can independently verify what you're doing. It can be very important for regulatory agencies to have access to the individual level data in order to verify the analyses. But we also recognize that there can be challenges with doing this regarding data ownership and other issues. But those are some of the things that help to make it easier to use this kind of real world evidence and regulatory decisions. But again, each case is unique, and we have to evaluate things very carefully in order to make sure that we're meeting all of our required standards when we're making decisions like this. Over.
That was extremely useful, Richard. Magda, do you want to add anything to this?
No, I just think no, nothing to add other just kind of acknowledging in terms of sort of thinking about sources, because there was your question sort of sources of data and who to trust and what to trust. It's a challenge, and I'm not sure that we've really solved it, right? And because there's sort of kind of layers of information as they get disseminated, and not just to, I'm just now kind of thinking kind of from a clinical perspective to clinicians, but then also to patients, right? Kind of the end user, all of this, who then comes to their provider asking questions, and their sources of information may be completely different. It could be, sort of Twitter and sound bites, et cetera. So how do we kind of, how do we ensure, and I think some of the conversations kind of touched on that also, how do we ensure that we as a scientific community kind of disseminate the information sort of very transparently in a nimble fashion, but that there is also kind of fact checking that that happens. And I think, so I don't necessarily have perfect answers, and I have to say that my sources of information have changed over time also. So I think kind of recognizing that we, as we validate the trustworthiness of certain sources, that we can also ourselves be nimble enough and adjust. I think that's also important.
Thank you so much, Joanne. Magda, sorry. Okay, so I think we are gonna ask George to help us a little bit to bring in questions from the community. In the meantime, if you want to think about additional items you want added to your wish list for the future, that will be appreciated, and we can review that before we close. George, do you have any good questions for the panel?
Yes, thank you, Danny. Thank you to the panel. So this is spectacular, and thank you for the support for OHDSI. So this has been wonderful. Before I begin, first, let me just say, please, to the community, ask more questions. We don't often get a panel like this assembled, so this is the opportunity. Second, I just want to ask a clarification. There's something you mentioned that hadn't occurred to me that how OHDSI could do this. So Magda, you mentioned biomarkers. Are you talking about things like antibodies, correlating them with infection rates as a baseline to then use antibodies going forward?
Perhaps, yeah, and even sort of thinking maybe certain markers of adverse events, right? If there are sort of biomarkers of just thinking about, kind of a super simple example would be myocarditis or pericarditis could be used as a marker. A troponin could be used as a marker of certain sort of cardiac injury. But even I'm thinking kind of from examples of in HIV, where in viral load, HIV RNA level was used as a surrogate marker of response to a particular medication, and it was also sort of being proposed as to be used as a marker of response to maybe certain kind of prevention interventions, et cetera. So that was just sort of that comment. And I think, Catherine, you had,
yes,
if you recall, yeah.
Catherine, please.
Yes, thanks. I just wanted to follow up. I totally agree with what Magdalena just said, and it was actually on my wish list for the closing discussion to have more linkage with laboratory data, with biobanks, to be able to look at also from, I have the term adversomics in mind, to be able to look at causality as well when we are assessing risk of some particularly complex events that can only be characterized using lab data or even pharmacogenomic data, but maybe that's a very not yet realistic wish. Thanks.
Thank you. All right, so now let me go through some questions from the chat. First, to Catherine, I think just very briefly, our audience is from around the world, so some of them don't know that much about the EMA. The question was, what are EMA plans around real-world data? How are you using real-world data in the epidemic?
Oh, that's a very broad question.
Yeah.
So okay, let me try and be synthetic. So actually, and this is relatively recent, I guess, in the mandate of EMA. Traditionally we were not conducting studies, but there are a few occasions where actually we need to generate our own data, particularly in a crisis situation where data has to be generated very quickly. So we have worked with the OHDSI network. I mentioned earlier the generation of background rates. We also have studies ongoing, and there are other situations. And I think I mentioned earlier the high media attention and some politically or from a public health perspective sensitive situations where we have to generate our own data. So there are a few databases that we have directly access in-house. We are also collaborating with networks, so OHDSI, also with some people might be aware also of the work we did with Access, with the EU Pharmacoepidemiology and Pharmacovigilance network. So we, on the one hand, we have external collaborations, we are funding studies, and we are using our own data sources. I mean, our own access to data sources. So it's complementing what is required from companies, and it's also used to input in for example, PRAC discussions. So I think that's about it. And of course, we will develop as we go forward the Darwin network in Europe, which will be a federated network of databases. So many different data sources in Europe and elsewhere as well. And this is only starting. Thank you.
Okay, thank you, Carol. The next, I'm going to combine the next two questions. One of them you may or not be able to answer. It's basically limitations of the data mostly on the US side. So this is more for Rich and Joanne. One example is missing inpatient medication administration data from US claims databases. Our Columbia database has it, but the claims databases do not always have that information. And also if you know it well enough, the OMOP common data model, are you seeing any limitations? So I'll just put those two together. Any comments? So I could make it a more general question, limitations on the data sources. Rich, you want to start?
Sure, George. The biggest limitation that we have faced in the United States so far is that we don't have good data on who has been vaccinated. And George, you talked about this in your presentation. In the US, especially the early part of the vaccine rollout, much of this was done in mass vaccination clinics. And while we had worked very hard to make sure that there were administration codes in place, that people could submit and be reimbursed for the administration of the vaccine, a lot of that didn't happen at the mass vaccination clinics. And so when you're trying to especially do an effectiveness study where you want to compare a vaccinated cohort to an unvaccinated cohort, right now we just can't do that because the group that appears to be unvaccinated is a mix of vaccinated and unvaccinated individuals, and we just have a lot of misclassification. This also has some problems for safety studies as well. Oftentimes, it can be helpful to do a cohort design for a safety study. We've been limited more towards self-controlled safety studies or comparison to background rates, such as we were doing in the rapid cycle analyses. So Overall, that's been the biggest limitation that we've faced. And I think it was Magna earlier that raised a question about biomarkers in these data. In the United States, our claims data, we might be able to tell if a test was ordered, but we won't be able to tell what those results were, except in rare instances where you've got an integrated system with electronic health records. And so that's one limit that's specific to the COVID-19 pandemic, and another that is a general issue in claims data in the United States.
Joanne, thank you.
Yeah, just to add to what Richard said, you know, understanding who really is unvaccinated is a real challenge. And I could say from personal experience, I'm myself volunteered as a vaccinator during, you know, when it was very busy. And you see that people who come to get vaccinated don't, in the mass clinics like the one I worked in, they don't collect insurance information. So going to the traditional databases doesn't always help us in terms of who is unvaccinated, because, you know, we were all trying to do things as quickly as possible. The other thing that I think is a general issue, but in epidemiology and the kind of work we do, but I think it's really been heightened during the COVID pandemic, is the data time lag. You know, really, we want to know what's going on during Delta, or we want to know, you know, the comparative safety. And if there's a big time lag in accessing the data, if the uptake of our vaccine is different than another vaccine, you know, there were different types of people who got the vaccine initially, then there, you know, or the numbers are lower until they get higher, you don't have enough. And then you have that data lag, and you have a critical question to answer, either about safety or effectiveness. And, you know, you're looking back at, you know, now in COVID, right? You know, a month is like a year, you know, in time, in terms of how quickly things are changing. So, you know, there are many challenges, but I would say that that's one of the things that has really been heightened during the pandemic.
Okay, thank you. The next question relates to OHDSI being an international effort, and so bridging all the most of the continents. We briefly had one person in Antarctica, just for the sake of saying it, but he moved out and his successor didn't answer his email. However, so we're back down to 6 continents. Is there anything OHDSI can do further to bridge the research that occurs, say, now we have on the panel representing very well US and Europe? Anything further OHDSI can do to help out? I don't know if it's bridging the 2 sets of databases, repeating the data, like, I don't know, what kinds of ideas do you have that OHDSI could serve a role there?
Yeah, I'm not sure I'm the best expert to answer the question in terms of what to do, but I'll just add that I think this is incredibly important. The vaccines, especially, you know, our vaccine is having a global rollout. You know, for example, Africa, South Africa, and many other parts of the world. And it's critical to have data everywhere in the world where the vaccines are being used. So the more representation we could get globally, I think the more benefit there will be globally in terms of both effectiveness as well as safety. You know, there are different concomitant medications, there's different medical use and medical experiences around the world. And COVID is different around the world, right? The variants are different around the world. Social behavior is different, right? Masking, even, you know, for those of us who live in the United States, it's different by city and state, but you know, it's vastly different by country. Social distancing can be done, can't be done. So I think the more global exposure to data analysts, people that we get, and the more experience we can understand globally, the better we will all be.
Yeah, and if I can, sorry, Catherine, go ahead.
No, go ahead, Magdalena, I'll go next.
Yeah, no, I was just sort of reflecting on where the OHDSI chapters are located, and I think Joanne, you were talking about this, kind of making sure that there is, you know, they're located in you know, certainly Europe, Australia, and Asia and Southeast Asia, and then I think Africa would be one you know, huge area where I think there would be great to be able to focus efforts on, and I understand that there are challenges also with, we're talking about kind of, collecting observational data in resource diverse settings, right, where maybe the sources of the data would be very different. And I was actually sort of wondering, but that can be also true in certain parts of US where there is no access to good electronic medical records. So I think a lot of lessons are translatable to these other settings as well. But I just think this will bring additional set of challenges. But I think it's critically important, as you were saying, Joanne, just as worth kind of thinking about kind of diversifying the population that we have the data from in order to make this information generalizable to large swath of the population, because this is and figuring out how to do it now, because COVID, it's not never going to completely go away. It will be sort of bubbling up every once in a while, and there'll be other kind of emerging infections that we'll have to tackle. So it's I think it's important to kind of be thoughtful about this now. So I don't have any answers.
And just to follow up on that, I think there's another important component is the sample size as well, because we are dealing sometimes with extremely rare safety outcomes, and of course there's already a quite large sample size available, but we might need more. So in terms of numbers, but also in terms of diversity of the data sources, and because again many of those also very rare outcomes are also only captured in secondary care. So more hospital data and this type of data. Thanks.
Danny, should I ask one more question, which will combine 3 of my chat questions so that I can, because we have 10 minutes.
That sounds like a good effort to me.
The additional wish list. It has to do with OHDSI's openness and trust. We put all our results online immediately before we see it. Then we look at the diagnostics, and then we look at the answers, and that way we can't change our minds halfway through. We don't, everyone, it's already published before we see the diagnostics and are allowed to say whether it's good or bad. So that's a good thing. But then in addition, as I gave an example in my own talk, that sometimes we do want to go to the chart. So, we're open, yet there's still limitations because our vast databases do have limitations, but we have some that are more deep. So what are the aspects that, basically, what do you need from us to improve the trust of our data? I mean, you could highlight the things that we're already doing, which I think you did at the beginning, which is, publishing on the internet, but there are other things that we need to do to improve trust of our results.
So George, I'll start by saying I think OHDSI is doing a lot of things right already, and I want to make sure to commend the OHDSI group for putting such emphasis on things such as posting protocols and being transparent with results, and making sure that that things are reproducible. I think it's hard for me to say precisely what other things would help. Having greater confidence in the underlying data is something that would help, but that's not something that's easy for people to do from a top-down perspective. But I think you've done a lot of the important things. Some of the foundational questions of whether the data that we have are appropriate for the questions that we need answers to, that's going to remain a difficult challenge. Over.
Other comments on that?
I was going to save this for my final comments, but I'll say it now. I agree with what Richard said. I would go a little bit even further. I would say that a lot of what OHDSI does is really being a role model for what needs to be done, not just in epidemiology, but in science. Transparency, openness, pre-posting protocols, but also questioning yourselves, right? Interrogating the methods, you're generating results, but at the same time, you have entire efforts on looking at the methods and questioning, are these the best methods? Let's look at all the available methods. Let's look at the differences across databases. And I think that the work on results and answering important medical questions, but also the work on methods and interrogation of what the right methods are in different circumstances, as well as the transparency. To me, I think that is kind of a role model for how we should be doing science, and especially epidemiology. And I really commend the community for doing that. Keep doing that, keep challenging yourselves, and keep generating, again, important information, not just the medical results, but also on the methodology research.
So just I couldn't agree in terms of kind of the kudos to the group sort of for being so thoughtful and kind of examining sort of everything that you've done, and sort of and asking these questions. And also I think just to kind of add another layer to it, not only how to present data quickly and transparently and accurately to the medical community, but then also maybe even sort of partner with the clinicians and with other scientists about how to translate these data to the lay audience, and especially kind of the some of the methodology, which is so beautiful and so important, how to, and maybe a little difficult to explain, but explaining it to the audience in order to kind of lend further validity to the real world and to the importance of these observational and real world data that essentially are amplifying and supporting clinical trials and lending kind of further and sort of and helping an uptake of certain interventions and answering a lot of fears and concerns. We talk a lot about speed and how, and there's pressure to publish quickly because information is changing so quickly, but kind of the general public oftentimes is mistrustful of that speed with which information is coming at them. So then kind of having thoughtful and transparent conversations about how these data were arrived at from the real world, and sort of talking about the methodology in an easy to understand fashion, I think is also important. So I think there can be some exciting opportunities there for partnerships also.
Very good. Danny, I had better turn it over to you for the final round. If there's any further comments, you can put it as part of yours.
Thank you, George. So I'm going to try and summarize very quickly the notes that I have on our Christmas wish list, which includes for now data on waning and need for boosters. I'm talking about COVID vaccines, obviously, effectiveness against different variants, validity of or exploration of surrogates for effectiveness in RCTs, and finally that very important point from Magda on patient involvement and PPI, as we call it, and how we transfer that knowledge to the general public. With that already ambitious agenda in mind, can each of you tell me, just to close up, any final remarks or any additional bullet points to add to that wish list? We can do this in the order we started, yeah.
I think we went through everything in my wish list, at least. So all I can say is I think it's really important to continue building capacity and excellence beyond COVID-19 vaccines for, prepared for the next public health crisis. Thank you.
Thank you so much, Catherine. Richard?
Yeah, so I'll add one more thing that I noticed during the presentations. It was fantastic to see such a talented group of people at the start of their epidemiological careers. And I think the community that you've built and the training that you're providing to help make sure that we have the next generation of epidemiologists that can help us is one of the things that you're doing very well, and that I hope that you continue with.
Thank you so much. That's an aspect I'm particularly proud of, although I'm still quite young. Joanne?
Yeah, I'll just add, keep doing what you're doing. Again, methods research, important medical research, transparency, openness. And I would just add, don't be afraid to be wrong. You see a database that has a different result, that there's something really important there that it's telling you, this is This is the time to self-reflect and interrogate the data, understand the data, and this is incredibly important for this. This is what OHDSI was built for, right? That what we're going through right now. So just a huge thank you for being role models for much of the scientific community, especially in epidemiology.
Thank you, Joanne. Magda.
Yes, no, I agree with all the remarks and kind of adding my own things. Just one other little wish list is sort of thinking also kind of about the real world effectiveness of certain mixing of vaccines and certain combination of vaccines of delivered through different, through different platforms, I think, because that will come up and has already come up in the real world. So I think that's also kind of an exciting and really important focus. I think that would be great to look for. And then again, just adding my own thanks. And it was really exciting to see a lot of junior investigators. So this just makes me happy because I know that whatever comes our way next, we're prepared. Thanks to you guys.
Thank you so much, Magda. Well, I'm gonna finish thanking you so much, all of you, for your time and for your contributions. It's been wonderful. I had a little bit of anxiety at the beginning with such a panel, but it's been great. And yeah, thank you so much. And hopefully the next generation is learning from this and will do things better in the future. Thank you so much, everyone.